Hey guys, I hope the Poggle went well the other day, especially if I wasn't there to help you. If not, hopefully this PowerPoint will help you clear some things up. I did have a set of notes for this PowerPoint that was printed out, but because I just changed the PowerPoint and I haven't had a chance to retype the notes, I'm not going to give them to you. I would suggest practicing taking your own notes from a PowerPoint without that guidance. Please do not write everything down. There isn't any need to do that. I am not going to read every single word, and so you can kind of listen to the cues that I'm giving you when I'm talking as to what is the most important information that you should write down. So we'll start with the cell cycle and how it's controlled. This slide just has the basic essential knowledges that are listed in the AP objective. So if you're going to try to get a 4 or 5 on the test, these are the kinds of things you have to be able to know. And then as far as what you have to do on the AP test, they want you to be able to make predictions about the cell cycle and then describe the event. So basically you have to be able to talk about what's going on. So starting with a diagram with all the phases, basically the cell cycle is the stages of a cell's life, and that includes interphase, which is this whole section right here. Everything basically that's not mitosis. That's going to include G1, S, and G2, and then M phase, which is mitosis or cell division. This is a nice drawing. I like these that explain what's happening in each one. If you're very visual, it wouldn't hurt you to draw this one or something like it. There's some great diagrams in your book. So then in terms of what's happening in interphase, it's got 3 phases. G1 or gap 1 is when it's growing. S is when it's synthesizing or copying the DNA. And then G2 is the second gap, and that's when it's preparing to divide or copying the organelles. I find that if kids forget a phase, they don't know what's happening in one, it's usually this last one, because it just seems maybe less important, but it isn't. So then how is the cell cycle or the division generally controlled? So you're using those checkpoints that you were reading about in the Poggle. If the conditions aren't appropriate, then you're not going to have division, and if you have too much division, then you have cancer. And at the end of the presentation, I talk more about that. It's really interesting. There's some interesting experiments about how we figured this cell signaling thing out. And basically, we know that there's these molecules present in the cytoplasm. They combined 2 different cells into a single cell, and those 2 different cells had been at 2 different phases of the cell cycle. And they found that the signals from one of the cells could cause the nucleus of the second cell to kind of go into the wrong stage. So for example, with experiment 1, we have one cell that's in S, where it's copying the DNA, and one cell that's in G1, where it's just growing and doing normal cell functions. And it turned out that the cell that was in S caused the second cell to also move into S before it was ready. So that tells us that there must have been some sort of chemical present in S that present in this cell that caused the cell to move into S. And the same thing happened with this one. We had a cell in M and a cell in G1. When we combined the 2 of them, they found that the second cell moved into M without copying the DNA, so before it was ready to do that. And so therefore the conclusion is there must be some sort of chemical, some sort of molecule present in the cytoplasm of those cells that would cause them to move into these different phases. So as we talked about on the day of we started the Poggle, the controlling the cell system kind of functions like a washing machine. You've got all these different timers. In addition to there being these internal controls that were talked about in the Poggle, there's also external controls, and we'll get to that in a minute. So these basically kind of function like a stop and go light or stop light, whatever you happen to call it. You don't have to memorize the different kinds of checkpoints and where they are and what they're looking for, but I like this visual. You notice it's happening later in the stage. That's the kind of Pogel pointed that out. So G1 checkpoints happening at the end of G1, the G2 checkpoints happening at the end of G2, M checkpoints happening at the end of M. If a cell doesn't receive the signal to move into the next stage of the cycle, it can go into that non-dividing state, which is G0, and that would look like this. And I don't think I talked about this with all classes, but G0 is a state that your nerve cells and brain cells are in, and that's why if you injure them, then you have trouble getting function back because there's no more mitosis, no more movement through the cycle happening, and whatever cell is existing at that point in your injury is oftentimes the only one you'll ever get. If you are younger and have a nerve injury, you're more likely to be able to recover from that because those are less likely to get stuck in G0 forever. So this graph should look kind of familiar. You were looking at one of the cyclins in the Pogel. Little did you know there's a whole set of them that control all different parts of the cell cycle. So you will see the word cyclin again, you will see the word kinase again, and you just need to know that there are chemicals that regulate the movement through the cycle. There are also external factors that can control the division. So the cyclin, the chemicals are internal, they're inside of the cell. And then there's external factors. One of those is called platelet-derived growth factor, or PDGF. And when you get a cut, your platelets, which are part of your blood, release this chemical that is external to your cells, and it basically attaches to them and says grow, and that's how you get new skin and other tissues grown onto your owies when you get them. In addition to the PGDF, another good example to know is the density of the surrounding cells. So basically, when a cell feels crowded, it's going to stop dividing. Another external control is anchorage dependence, where they have to kind of be attached to something in order to divide. What's interesting is cancer cells don't sense either of those external factors. So what should happen is you should, have this surface upon which the cell should be attached. It should be sensing what's happening around it and form like this single layer and, make new divisions to fill in any gaps. And that's what will look like in a normal tissue. Excuse me. But cancer cells don't have any regard for space. They're kind of like maybe the subway in Japan, from what I hear, where they get super crowded, and they also will divide without being attached to something, which makes them very dangerous. That's why some cancers can slip around your body. So that is how cancer cells ignore those external factors. They will also have ways of ignoring internal factors. They might be able to divide without the growth factors, like those cyclins. Sometimes they make their own. Sometimes they manage to say that it's time to divide without that factor present. And then sometimes they'll just have other different mutations within those signals that will cause them to divide whenever they want. Every cancer is different with this, and even within certain cancers, like let's take breast cancer, there's a ton of different mutations and different kinds of breast cancer that are caused by all kinds of different gene mutations and other issues. So there's too much to go into. There's a whole course you can take in college called Biology of Cancer, in which you can learn more about that. So before we go further into mitosis, I want to just clarify some vocab. These are not difficult words, but they're definitely confusing, and kids have had trouble with them in the past. So these are worth drawing out in a book. All right, so first, the word chromatin is basically a bunch of DNA and protein. Every organism is going to have a different number of chromosomes, which are made of that chromatin. You should know the word somatic cells. Somatic cells are basically any cell that isn't a sex cell, and those should have 2 sets of chromosomes. So your skin cells, your hair cells, your heart cells, stomach cells, all of those are somatic cells. You might remember, I think one of our word roots was som, it means body. So I think of body when I think of somatic cells. Gametes are a fancy term for sex cells. And in animals, that would be sperm and egg. In plants, it would be the pollen, which is like the plant version of sperm, and the ovula, which is like the plant version of eggs. And they are different than body cells because they only have 1 set of chromosomes. If you think about why that would be, you got 1 set from your dad and 1 set from your mom, which would give you 2 sets for somatic cells. You are not going to be dealing with gametes in the rest of the presentation. We'll do that after actually this first test in genetics. So the reason we're talking about this now is because part of cell division is having the DNA, copied in S phase and then starting to condense during mitosis. And when a duplicated chromosome or a copied chromosome that was copied in S phase is copied, it has these things called 2 sister chromatids, which are identical copies of the original chromosome. And they are attached with something called a centromere. So here would be the 2 sister chromatids, and there's the centromere. In drawings, it looks like a little dot usually. And then during cell division, those 2 sister chromatids of each duplicated chromosome are going to separate and move into 2 different nuclei. Nuclei is the plural of nucleus. Once those 2 sister chromatids are separate, we call them chromosomes. So I'll show you a picture of this. So this would be 1 chromosome that's not copied. So this would be like in G1. And then this would be another chromosome, or the same chromosome, when it is copied after S phase. Notice down here, they're still both considered 1 chromosome. I will refer to this as a duplicated chromosome with 2 sister chromatids. Although sometimes, like when I'm counting the numbers of chromosomes, it helps me to think about these as being separate chromosomes. And I'll show you guys that when we model this when I come back on Friday. So what would that look like in cell division? This would be, again, G1, where we have 1 chromosome that's not copied. And notice over here I just have 1 double helix. And then when I copy it, I have 2 sets of DNA and 2 sister chromosomes, 1 chromosome. And then when I separate them again, these are both considered 1 chromosome, no longer sister chromatids, and there's only 1 set of DNA in each of the 2 cells. So again, this is 1 chromosome, this is a duplicated chromosome with 2 sister chromatids, and these are both unduplicated, just regular old-fashioned chromosomes. Another term you have to know is homologous chromosomes. As a human being, we have 2 of each kind of chromosome. One of those homologous chromosomes is maternal. One of them is paternal. And they have the same kinds of genes in the same spot. Same kinds of genes in the same spot. So there's not actually ever really very good definitions of homologous chromosomes. So I think of them as a pair of chromosomes, one from mom, one from dad, that have the same kinds of genes in the same spot. That's my definition of that. I like this picture. It's something I found on the internet long ago, but it shows like the idea of same genes in the same spot. So for example, this could be a gene for curly hair, and because this is the sister chromatid on the other side of it, it is also the gene for curly hair. As you can see here, the sister chromatids are exact replicas of each other. So this sister chromatid is exactly like the sister chromatid. They just copied themselves in S phase to make identical copies to do mitosis. However, this chromosome may have come from my mother, and this chromosome may have come from my father. And it's very possible that they are going to have different, now I lost my spot. It's very possible that they're going to have different alleles. So this one could be curly hair, and then this could be straight hair. So same kind of gene, it's a hair gene, but it's a different version. This could be the gene that causes cystic fibrosis, the recessive gene. Both versions on both sides. Over here, I could have the dominant version of that, which is the gene that does not cause cystic fibrosis. Either way, both of those blue stripes have to represent cystic fibrosis. Just they could be different versions, or they could be the same. This is another good drawing that I really like of what homologous chromosomes look like. If we were to lay out all of your chromosomes as a human being, we call that a karyotype, just a word that you'll probably see. This is what that would look like. You have 23 pairs of chromosomes for a total of 46. And the last pair we call sex chromosomes. These are called autosomes. They're basically chromosomes that have genes for everyday functions, and sex chromosomes code for things like testosterone and estrogen and things that make us male and female. We will talk more about X's and Y's and issues that can happen with that at the end of the unit. Notice the difference between this picture and this picture. So in this one, I have copied the DNA. I have identical copies of every single pair. So if over here, I have 46 chromosomes, over here, technically, you still have 46 chromosomes, but because each chromosome is duplicated, I like to call that 92. Or you could just call it 46 duplicated chromosomes, as long as you can keep track of what's happening there. This would have happened after DNA replication, so this would be after S phase, but before the cell divides in mitosis or meiosis. As I said earlier, every organism has a different number of chromosomes, so this is a karyotype, like let's say for a mouse. By the way, there's a mistake here. You can see that the mouse has an extra 17th chromosome. That will happen in cell division, and we will go over that in a few weeks. And a puppy, because it's cute. Okay, mitosis. Again, if you are concerned with the specific things you need to know for the AP test, and don't trust me, here they are. And then for objectives, they say construct an explanation using visual representations or narratives. In other words, explain mitosis in writing or interpret a diagram of it. So mitosis would just be one part of cell division. It's the division of the DNA, and then cytokinesis is the division of the cytoplasm. Cyto means cell, kinesis means cutting, so you're cutting the cell. You can make sex cells using cell division, but that's meiosis, and again, we're not doing that right now. In meiosis, you would get non-identical cells, because we wouldn't want all of the sperm and all of the egg to be exactly the same, because that defeats the purpose of, or part of the purpose of reproducing through sex. And they also only have half as many chromosomes as the parent cell when you make sex cells. In mitosis, we are going to make two genetically identical daughter cells. So if you're going to write one thing down, that would be it. They have to be exactly the same. You're supposed to know that the one skin cell that you're going to scratch right now on your hand is going to fall off could have divided into two exact skin cells, just like the one you just had. They should have the exact same kinds of DNA in the exact same spot. Basically, mitosis, all there really is to it is that the DNA lines up in the middle like this, and then it pulls apart, and then you've got two cells that look exactly like the very first cell that you started with. There are 5 phases. You technically do not have to know these. I kind of find it helpful to know them, because then I can use them to describe it later on. Like, just having a title attached to the different descriptions is useful for me, but you don't necessarily have to know that. Supposedly, that's what the College Board says. I also don't 100% trust that, so just learn them. Okay, here we go. So, first phase of mitosis is prophase. They are showing you in this diagram the end of G2, though. Notice that the chromosomes are not condensed here. It's just kind of exists as that chromatin. So it's just kind of this loose, all over the place DNA. And in the very first phase of mitosis, we're condensing them. Kind of thinking about having like a big pile of yarn that your cat unraveled. That's what this is. And then in prophase, it's like you're wrapping that back up into a nice little skein of yarn that your mom will use to knit you a sweater. And notice how this is doubled, by the way. That happened in S phase. And then in prometaphase, the nucleus starts to disappear. Notice that little purple piece, like over here, that was this thing. And now it's almost gone. And that needs to happen so that this can happen. So if on a test I was to ask you, when would the nucleus disappear? And I had like some pictures maybe laid out of, you know, the order, like I labeled them prometaphase and metaphase, etc., but I didn't have any drawings in there. You would know what happens earlier in the process, but not when the chromosomes line up. Like, this breaking apart has to happen first, kind of like releasing the chromosomes from a little jail. These little tubules or sometimes they're called spindle fibers. They're technically different things, but you don't need to know that. They're basically little fibers that reach out and grab onto the chromosomes. And notice that they're attaching at the centromere there, or somewhere around there. I remember that metaphase is when they line up in the middle because M for middle. And then at anaphase, they're being pulled apart. And I remember this because it looks like an A. And then lastly, in telophase, there is cytokinesis starting to happen. Notice a new nucleus is forming around here. And notice how I only have one of each chromosome. So in a human, at this point, I've got 46 duplicated chromosomes, or you could think of them as 92. And then down here, I only have 46 in each. So this is a series of pictures of plant cells dividing. They look a little bit different. Mostly here at the end, when this cytokinesis is happening, forms this thing called a cell plate. These slides have, text on them about what's happening. They're probably a little bit too detailed, but I kind of like the diagrams. There's a really good video, it's only 3.5 minutes, that I would like to show you, but it has sound and it sounds crappy when I'm recording it. But I am going to show you this one without sound, just because it's super cool. So this is a real animal cell dividing. And right now, the chromosomes, like, they're condensing, and the nucleus is probably breaking apart, and they're starting to try to kind of line up in the middle as the nucleus breaks. So you can't see the spindle fibers reaching out to grab them, but that's, that moment. Here's metaphase, then boom, you've got anaphase as they pull apart. Telophase and cytokinesis as the new nucleus forms and the cytoplasm is cut. And you've got two identical cells, like magic. Cytokinesis is, again, just when you're cutting the cytoplasm, it will look something like this in an animal. Sometimes this space is called a cleavage furrow. And then in plant cells, again, it forms this little spot called a cell plate. So that's essentially it for mitosis in terms of phases. Again, you don't have to memorize them, but it might help to draw them out. Please don't obsess over them too much. If you're going to have a weakness, it'll probably be in the protein synthesis stuff, so that's what you should be studying more fervently. All cells reproduce using division, but in prokaryotes, we call it binary fission. So what we just went through would be for eukaryotes. Remember, prokaryotes have circular DNA. That was like the first thing I taught you in the genetics unit. So that's why these are showing nice little loops. Remember, they also have these extracellular pieces of DNA, which they can kind of pass between each other, and those are copied as well. So notice each of these two cells here has a plasmid. What's interesting is we think that mitosis exists because prokaryotes did something similar to that. So mitosis in eukaryotes would have descended from prokaryotes that were doing binary fission. So the end of this presentation has just a few questions just to make sure you understood. So let's go through these. So how many chromosomes are there in the middle cell? This one. Are there 1, 2, 4, or 8? Okay, hopefully so you said 1, because even though it has 2 sister chromatids, we consider that to be 1 duplicated chromosomes. If you had to count chromosomes, which you might on a test, you would call that 1. So if you were given a picture of this, what part of the cell cycle would you think that it is in? G1, G2, M, or S? So I want you to think about how many chromosomes are present, or how many sister chromatids are present. And I want you to think about the shape of them. Are they stretched out, or are they condensed? So hopefully you're realizing that they are condensed, and that there's 2 sister chromatids. So the fact that there's 2 of them means it must be after S phase, which would leave me with G2 or M. But I know that it's M because at the beginning of M, at the beginning of mitosis, I condensed the chromosomes in that phase called prophase. If it was G2, they'd be duplicated, but they wouldn't be condensed. So they won't look like this nice wound up. They look like that messy ball of yarn or a bowl full of spaghetti. So although the chromosome begins M phase looking as shown below, when does it split? Can you remember what phase of mitosis is the splitting phase? That would be anaphase. Remember, they look like an A as they pull apart. And then if you were to examine a sample of connective tissue from cells from a small but adult mammal and a second sample from a fetus of the same species, in which would you expect to find more cells undergoing mitosis? So from a fetus or from an adult? Hopefully you're realizing that it would be from a fetus because they are having to grow very quickly. It's the same thing, interestingly, with plants. So if you look at the bottom of an onion versus like the top part of it, the bottom is where, I should say, the bottom of an onion root versus the top of an onion root. Roots grow from their tip. So the bottom of the onion root, like the part that's deepest into the ground, has a lot more mitosis happening in it. One thing we'll do on Monday or Friday when I get back is we'll just look at a picture of some different cells in different phases of mitosis, and there'll be onion root cells, and we'll kind of identify the different phases that they're in. Okay, I hope that was helpful. Please look on Blackboard for the rest of the directions for today.